before starting. Recording in progress.
Dear colleagues, today is the day of our Kharkiv chemical seminar, and our guest, Professor Remi Chauvin, who is a big friend of Ukraine and Kharkiv city as well. And Professor Remi Chauvin studied at National School of Chemical Industries and National School of Chemistry in Paris, where he received a bachelor's degree in chemical engineering. He pursued a master's degree and PhD at the University of Paris-Saclay under supervision of Professor Chauvin. Afterwards, he worked as a postdoc, first in the group of Nobel Prize winner Professor Barry Sharpless at the Massachusetts Institute of Technology in the USA, and then in the University of Zurich in Switzerland. In 1996, Professor Chauvin received his habilitation at Paul Sabatier University, Toulouse. And for 24 years, he was a research group leader and director of the Laboratory of Coordination Chemistry at the University of Toulouse. From 1998 until now, he has been a professor at the University of Toulouse. From 2015 to 2020, he dedicated 3 months each year to research and teaching in Huaqiao in Xiamen, China. In 2023, Professor Remi Chauvin spent 3 months conducting research and teaching in the National Autonomous University of Mexico. His research interests include the study of carbobenzenes with various physical properties, the development of anti-cancer and anti-tuberculosis compounds, the investigation of electrostatic effects from polar phosphorus ligands in catalysis, and theoretical studies of the structure of organic compounds. He has received numerous awards, but among them, it's very important for us, a Medal of the National Academy of Sciences of Ukraine in 2019, and he is also a honorary doctor of the Institute for Single Crystals of the National Academy of Sciences of Ukraine, Vasil Stus Donetsk National University, and Taras Shevchenko National University of Kyiv. So, dear Remi, please, you can start your lecture.
Thank you very much, Valentin. Actually, you mentioned everything, so you make me save time about the next slides because you presented some points that, okay. So first of all, I thank you for the kind introduction and very complete actually, and also for the invitation. I can only be sorry to not be present in person today in Kharkiv, but make sure that I don't forget the Institute for Single Crystal. I don't forget you. I don't forget Kharkiv. I don't forget Ukraine and all the friends I have in Ukraine and of whom I think every day. I think of every day. Also, I would like to express my pride, as you mentioned, to formally belong to several research and teaching institutions in Ukraine as a doctor honoris causa, as you can see, of the University Taras Shevchenko in Kyiv, of the University Vasil Stus, Donetsk Vasil Stus University in Vinnytsia, and of course of the Institute for Single Crystals since 2019. Actually, you can consider, this talk can be considered that after 5 years after my graduation, okay, from the ESC, okay, this talk will be kind of official honoris causa talk, okay? This is kind of introduction, okay? And please, I will, I cannot go. Where is the next slide? How do you change the slide?
Just
okay, yes. Okay, now sorry, I can see now. Okay, sorry. Actually, you summarize very well. So our research activity over the last 25 years. Okay. However, here is a kind of triptych of the group's research, and the group is entitled, okay, Unsaturated Molecules for Physics, Chemistry, and Biology. So it is a very eclectic research activity. We are not expert in any of those panels, the 3 panels. However, we focus on on specific properties of 3 kinds of molecules in 3 different scientific domains, like carbomeric molecules for physical properties, polar PC ligands for catalytic and chemical properties, and of course, lipidic alkynyl carbinols, the so-called lipidic alkynyl carbinols for biological properties, and especially anti-cancer and anti-tubercular activity. So actually, I will develop today, I will focus on this panel of the triptych. So actually, the original outline in my abstract consisted in 3 parts, okay? I was very ambitious, okay? And speaking about anti-cancer lipidic alkynyl carbinols, okay? Then related molecules against tuberculosis, and also on a new kind of molecules directly inspired, which are anti-cancer dacamines molecules. However, due to the lack of time and due to actually some thinking, okay, I could not speak about everything in 45 minutes. So finally, I decided to refocus from the abstract. So the abstract I sent you is on the screen. And so I will not speak today about the aspects highlighted in green, namely tuberculosis, lack or alenac diols, and the dacamines, okay? I will focus mainly on the aspect highlighted in orange color. So first of all, so the today's outline is this one now, okay? Just focusing on the first part. So first of all, where do they come from, the lipidic alkynyl carbinol? Actually, in the first panel of the research triptych, we focus for a long time on acetylenic chemistry for material properties. And from material property to natural products, it was kind of a small step to go. Actually, carbobenzene have many properties, physical properties. One is depicted on the left. It is a carbobenzene, and carbobenzenes are prepared from 6 pericycline diols at the center of the slide, which actually contains 2 secondary dialkynyl carbinol units. Okay, and the acronym for this, the abbreviation is DAC. The acronym is DAC, dialkynyl carbinol. So those highly functional C5 units are, of course, very reactive, okay? And we wondered very early if it could be found in nature. And actually, polyacetylenic natural products are relatively rare, and it was maybe tempting to check if such a motif could be found. And indeed, Viviane Peroux was my first collaborator working in this field. Okay, that was in 2004 and 2003, actually. And she found in the literature that lipidic derivative of DACs, okay, were extracted from marine sponges, and especially various petrocortines, which display at the central in the center of the structure a DAC motif. Okay. And actually, it could be shown by Shin and Jung, okay, so Korean researchers, okay, that those molecules exhibited some cytotoxic activity against human tumor cell lines, okay, and very soon, not very soon, but sometime later, Chauvin performed the total synthesis of these, of 1 or 2 of those petrocortines. Actually, the petrocortine contained 2 motifs, a DAC motif and an alkynyl carbinol motif that we call the AAC motif in blue, okay? And we wondered which one was the pharmacophore of the biological activity, reported biological activity. Actually, the same 2 motifs can be found in fulvines, which are extracted from marine sponges, okay, not in the Korean Sea, but in the Mediterranean Sea, and they have been reported to be to have antimicrobial activity. And also, we can find in the literature other lipidic AAC molecule, C2 symmetrical, okay, without a DAC unit, okay, from Adosia species, okay, and it is called durines. And the durines have attracted also a great deal of interest regarding their biological, their extraction, activity, and total synthesis. So actually, in the search for simplicity, it was possible to find a molecule extracted from a marine sponge, which is a Tuberculosis vasculum from the Caribbean Sea, okay, which has a single motif, a reactive motif, namely the AAC motif, okay. It was found in various enantiomeric purity in those various locations of the sponge, and the molecule has attracted interest, in particular for their anti-cancer activity against lung cancer. And of course, some efforts were provided by Chateau-Padois in particular for the total synthesis of the molecule. So finally, reasoning as chemists, okay, synthetic chemists, we separated the structure in 4 different sections, namely the carbonyl section, the central carbonyl section in red, and especially regarding its configuration, CIP configuration. Then, of course, the second parameter, structural parameter, is the chain length. So it is a lipid, of course, but how important is the length of the chain? And then, of course, the nature of the internal and external unsaturation, which can be double or triple bonds, and we wondered on the possible comparison between those molecules. Actually, this analysis suggested 72 candidates to be obtained by asymmetric synthesis for optically pure derivative. So finally, we tried first the synthesis of the natural AAC molecule extracted from Tuberculosis vasculum, okay, using the PUS oxypropargylation method, using diethylene zinc and binolate titanium, titanium binolate, sorry, and actually it was performed in using the described procedure. So starting from the corresponding aliphatic chain, using S-binol and R-binol, it was possible to obtain the corresponding natural products in 90% EE, and the opposite configuration was obtained in 80% EE using the enantiomer of binol. Then, of course, we changed the chain length, and for instance, with the dodecane chain, we could obtain the 2 enantiomers corresponding to the homologues of the natural AAC. Actually, the absolute configurations, which are shown here, could be determined by X-ray crystal structure of a carbamate obtained by condensation with naphthalene isocyanate, okay? And actually, the enantiomeric excess was confirmed by supercritical fluid chromatography, and as you could see in this slide. So then we wondered about the same kind of the same kind of synthesis for the DAC unit. Actually, we used the modified version on which we have been working a few years before in 2006 for another purpose. And of course, we tried to obtain the DAC by asymmetric synthesis using the modified Carrera's method, modified but simply by changing toluene by dichloromethane and involving 4 equivalent of the chiral inductor, which is just N-methylephedrine, okay? And of course, so we could actually check the relative reactivity of the lipidic chain and of the nucleophilic part and the nucleophilic part of the method, okay? Using, of course, still zinc triflate, which is a very convenient metallic reagent, okay? And we could see that finally the optimal combination is achieved with 90% EE roughly, using the lipid part as the nucleophile towards the propionyl, which protected propionyl, which is the electrophile. So it is still the step of the introduction. So now, what about the absolute configuration? The absolute configuration, according to the Carrera's rules, okay, could be anticipated to be the corresponding R configuration. However, it could be checked, okay, by Viviane Peroux and by Dimitri Listunov. I will speak a lot about his work in the next slides, okay, by derivatization with mandelic acid, okay, and it could be indeed confirmed that the SS configuration was obtained with the opposite enantiomers. So don't worry, it is completely consistent with the Carrera's rules. So then, after using those two methods, okay, we started a systematic structure-activity relationship with respect to the study, with respect to the anti-cancer activity against HCT cancer cells, okay? And actually, it could be shown that starting from the natural AAC extracted from Cribo-Alcalina vasculum, by changing the configuration from S to R, okay, the activity, the IC50 value against HCT116 cancer cell line, very standard, okay, was multiplied, the activity was multiplied by 5, namely the IC50 divided by 5. Then changing the chain length, okay, shortening the chain from 14 to 11, and 15 to 12, okay, then another enhancement of the activity by a factor of 5 was obtained. And finally, changing the internal insaturation of the AAC, natural AAC, which is a double bond, to a triple bond, we could obtain artificial DACs, okay, with another enhancement of the activity by a factor of 5. It is noteworthy that the terminal DAC here, not at the center of the molecule, and as in petrocortine, is today not found in nature. Okay, maybe one day, but it is an artificial molecule, definitely. So then we performed further structural variation. I will not go too much into details. However, I would like to mention that beyond, of course, the chain length, the absolute configuration, and the nature of the insaturation, internal insaturation, the possibility, for instance, of fluorination around the pharmacophore, or the introduction of oxygen atom on the up to PEG, actually, and polyethylene glycol chain on the aliphatic chain did not improve the activity. And what is important here is actually that changing the carbonyl unit, secondary carbonyl unit, to the corresponding ether, or changing the secondary carbonyl unit to a tertiary carbonyl unit, or oxidizing the secondary carbonyl unit to a keto group, dialkynyl keto group, gave very, very low anti-cancer activity against HCT116 cell line. So finally, we performed an optimization of the chain length in the racemic DAC series, and as you can see, the optimal chain length could be determined to the value n equals 12. So for n equals 12, corresponding to the C18 backbone, the automeric and the distomeric enantiomers of the corresponding DAC could be obtained by the classical modified Carrera method, and we could obtain IC50 value for the eutomer of 16 nanomolar, and for the distomer it was 1000 nanomolar. So as you can see here, so finally it confirms the intuition. And noteworthy is a quite high eudismic ratio of 17. Then we wondered about what is the target, the cellular target of the DAC molecule inside the cancer cells, the HCT116 cells. Actually, we designed a bifunctional molecule, okay, with the pharmacophoric unit, DAC pharmacophoric unit, and of course a fluorophore, which could allow, of course, exploration, monitoring of the cancer cell, okay, by confocal spectroscopy. And actually, so we performed the synthesis of the triene, which is a DAC-ine, where the terminal triple bond is added at the end of the aliphatic chain in order to perform click chemistry, okay, and of course to have the fluorescent motif, allowing the localization of the molecule after treatment. Actually, the DAC-ine precursor was synthesized, and it could be obtained in enantiomerically quite pure form in 95% EE or 97% EE, depending on the protecting group. It was obtained using the classical modified Carreras method. Several fluorophores were anchored by a click chemistry, so in lab, okay? And of course, the fluorophoric unit were derived from coumarin, from body P, and from nitrobenzoxadiazole, NBD, okay? And the molecules were fully characterized by their absorption and emission spectra. So after that, we performed with Etienne Joly, so the confocal fluorescence microscopy as planned. And as you can, of course, sometimes a biphoton excitation was necessary, especially for the coumarin tag. And here, as you can see, both the three tags, B, C, and D, namely coumarin, body P, and NBD, show that the targets are intracellular membranes, which are correspond to the endoplasmic reticulum, okay, ER. And actually, the DAC-ine precursor was also envisaged to undergo click chemistry, but not in vitro in the lab, but in cellulo. And that was for the determination of the proteic receptor in the cell, using classical biotin conjugation with the fluorophore and the streptavidin pull-down that was envisaged quite early by Sébastien Briton in Toulouse. And of course, very shortly after, so using no longer HCT11 cancer cell, but another class of cancer cell, namely U2OS, which are osteosarcome cancer cells, okay? He could show by fluorescence spectroscopy, okay, fluorescence imaging in cellulo, that using various dyes, and that actually the dye corresponding to our molecule, which was actually on the receptor in the cell, okay, it could show that it is around the nucleus and the mitochondria, okay, and it could show in the first line that only one enantiomer of the DAC was attached, reacted with the receptor, was so only the eutomer. The distomer did not display any coloring. So after clicking label with Alexa Fluor, okay, so and of course imaging at 488 nanometers, so by classical click chemistry, but in cellulo. And as you can see, this was quite I would say spectacular regarding the enantiospecificity of the the mode of action. So the mechanism of action of DAX, okay, was first attempted by Sébastien Briton on haploid HAP1 cancer cells, okay? And as you can see here, so he could compare the two enantiomer, eutomer and distomer, and indeed, effectively, the viability diagram shows that the distomer is very poorly active, more than 5 micromolar, while the eutomer is 62 nanomolar and active. So finally, after mutagenization of the wild-type HAP molecule and test with SDAC molecule, which was synthesized by asymmetric synthesis, it could select resistant clones, okay, resulting from this statistical mutagenization. And after that, finally, it was possible to, after RNA sequencing, to identify two kinds of genes corresponding to two kinds of protein, namely KCTD5 and HSD70B11, okay, which were mutated at the serine for leucine and the valine 16 for aspartic acid moiety. So after that, okay. After that, so a verification what actually all the other clones which were inactive, okay, deactivated, indeed exhibited HSD17B11 mutation, okay, and it was then shown that actually the HSD17B11 protein was absent, okay, from the resistant clone, okay, by immunoblotting, as you can see here, for the green resistant clones that are shown here. And of course, the HSD17B11 protein is almost absent, okay? So finally, the conclusion is that the SDAC cytotoxicity depends on one enzyme, which is SDR. SDR is a short chain dihydrogenase reductase enzyme, okay, which belongs to a much bigger, the family of which is very large, and so we had a good understanding of the mode of action. Actually, HSD17B11 protein is actually an oxidase, okay, converting the androstenediol to androsterone, okay, and it is mainly located, as suggested before, in the ER system and in the lipid droplets of the cells. And actually, it could be imagined that the lipidic chain of the DAC actually can be backfolded as a mimic of the steroid skeleton of androstenediol. And actually, that was checked by various experiments or outside the cancer cell. And actually, the proteins, several proteins can undergo lipoxidation through basic amino acid using those DACones, which could be the active species. Actually, the mechanism is a prodrug mode of action. And actually, what happens is that after a Michael addition, for instance, of cysteine, of course, a soft amino acid end, okay, so the protein are lipoxidated by the depicted way, and entails, and the lipoxidation entails inhibition of the ubiquitin proteasome and protein quality control of the cell, allowing which can be appraised by the endoplasmic reticulum stress, and which leads to apoptosis. And actually, this was checked with a particular protein, okay, nucleophilic protein, namely which is beta lactoglobulin, as you can see, which has a cysteine, which can attack in a Michael addition manner, so the corresponding DAC cone. And actually, after click chemistry with Alexa-Fluor 647 and electrophoresis and mass spectroscopic analysis, we could see that the lipoxidation of BLG had indeed occurred, okay, in vivo. So that was the definite proof of the product mode of action of the DAC. Actually, here is the docking, okay, and the docking of the molecule, and you can see that starting from the natural substrate of HSD17B11 protein, namely the androstenediol, if you optimize, okay, the receptor cell in the presence of the DAC, you have a almost perfect superimposition of the lipidic chain of DAC with the lipidic chain of the steroid, okay? And actually, this was a very, very exciting result regarding the mode of action. So now, those molecules are acetylenics. And of course, as I told you at the beginning, so alkynes are quite rare in pharmacopoeia, okay? There are some examples, there are several reviews about that, but quite rare. What about increasing the acetylenic character of the DAC by adding another triple bond in the so-called butadiene alkynyl carbinols, receiving the acronym BAX, the BAX. Actually, the modified Carrera's method could be applied to the butadiene's nucleophile, okay? Not only one triple bond, but two triple bond in red, okay? And actually, we could obtain the two enantiomers of the BAX, okay, in 85% E, so similar to the case of the monoine nucleophile. And actually, the cytotoxicity against HCT116 cells could be determined as 42 nanomolar for the eutomer and 1,500 nanomolar for the distomer with an odysmic ratio of 36. And of course, so the same rule regarding the Carrera's method of the Carrera's method for the addition nucleophilic oxypropagylation process could be confirmed by X-ray diffraction analysis. So actually, now the same was the same optimization of the chain length performed in the DAC series was performed in the BAC series. And in this case, we could see that there is a very sharp optimization with actually a minimum of the IC50 value for n equals 9, okay? So it was, in the case of DAC, the curve was much flatter, okay? And in this case, it is very sharp. So using the optimal chain length, we could determine, so we could synthesize the eutomeric BAC and the distomeric BAC in optically pure with the right chain length, okay? And as you can see now, with the optimal chain length, the IC50 value of the eutomer dropped to 15 nanomolar. So which correspond to roughly 700 times higher efficiency than the reference natural product extracted from Cribocalina vasculum we started from, okay? So that was a great improvement. It was a bio-inspiration, of course, but then artificialization, systematic artificialization led to a much more active molecule. And finally, we wondered, however, a big problem is that BAX, the BAX with butadiene unit, are actually very reactive. As you can imagine, you have three triple bonds in the vicinity of the secondary carbonyl unit. So they are very active, but very sensitive to storage. And theory actually suggests that an acetylenic ring, okay, namely a two-membered ring, is a 2-annuline, and it can be compared to 6-annuline, which corresponds to benzene. And so, starting from the back at the center of the slide, okay, and you have the extra possible extra triple bond added to the original DAC, and in green, okay, and we could wonder if it was possible to change the triple bond by a phenylene unit, and with the interrogation mark, which is shown here. Actually, the phenyl group could also be used to replace a red triple bond, okay, either external or internal. But in those cases, the synthesis were performed with the racemic series, and the IC50 values were much higher, showing a significant decrease of the cytotoxicity. So finally, we wondered on the synthesis and systematic studies of the phenyl dialkynyl carbinols, which receive the acronym PACs, okay, by two complementary routes. And in this case, actually, it seems that the lipidic chain should be optimally used as at the electrophilic side of the Carreras process, modified Carreras process. Here you can see it is the aldehyde electrophilic end, okay, bears carries the lipidic chain. And in both cases, so finally, the corresponding no, sorry, it's not the Carrera method. It's not asymmetric yet. Okay, sorry, it is just the racemic synthesis. Okay, and actually that was the reactivity of the lithium salt or the Grignard salt. Okay, and we could obtain the corresponding cytotoxicity of about 150 nanomolar. Okay. And by comparison to the corresponding DAC, it is roughly the same, because as you can see, with a chain length of with an optimal chain length, okay, it could be it was 180 nanomolar. And by comparison to the DAC, of course, it was much less cytotoxic, much less efficient. It's less reactive. And actually, so the process, which we actually relies not on the nucleophilic addition, but on the Sonogashira coupling, and then at the first step, and then on the nucleophilic addition to the carbaldehyde moiety. Then we could compare the activity of the PACs against HCT116 cell line, and compare it to the DAC series, as you can see here. So one of the molecules, because many molecules were synthesized, and one of the PACs was twice more active than the corresponding DAC. Okay, as you can see in green, here you have with the fluorination in a benzylic position on the internal aliphatic chain, give 90 nanomolar as the IC50 value, so quite active. And of course, also, several aromatics were tested, okay? And also it is remarkable that the corresponding furane derivative also exhibited roughly quite high cytotoxicity, because, not because yet, okay, there is no analysis of that, but that was remarkable anyway. So actually, the original idea was to stabilize the back, and the back system are indeed much more reactive, as I would say, suggested by the decrease of the IC50 of the increase of the IC50 value by varying the incubation time of the molecule. So in a protein medium, okay, in a complete culture medium, okay, as you can see, so there is a reactivity and which decreases the activity of BAC. However, the BAC is even more stable than the DAC, okay, with a decrease of a factor of 1.6 after 8 hours instead of 4.9 after 8 hours for the BAC series, and a factor of 2 after 8 hours for the DAC series. So finally, the asymmetric synthesis of the packs were performed using both the electrophilic and nucleophilic side of the acetylenic chains, what I wanted to say in the 2 slides before, okay? And as you can see, the highest enantiomeric excess was obtained with the nucleophilic part anchored to the electrophile. So it was the reverse of the optimal combination observed in the DAC series using the same method, modified Carrera method. As you can see, so the molecule could be perfectly isolated. And however, so as you can see here, so finally the fluorinated pack indeed under the eutomeric form exhibited twice the activity as the racemic series, so suggesting that the other enantiomer is just inactive, okay? So it is 42 nanomolar for the S enantiomer. So now I would like to finish with an update, recent update. Actually, the BAC actually are much more reactive, but much less stable. And the packs actually are maybe a little bit too stable to be biologically, to exhibit an optimal biological activity. Actually, coming back to the theory of aromaticity, both benzene and acetylene, acetylene, okay, as I mentioned before, are 4n+2 topologically aromatic system. It is very theoretical. However, we can wonder about the role of theo-inspiration by comparison to bio-inspiration in medicinal chemistry. And according to Breslow, dearomatization process, okay, standard, which was published in I haven't given the reference. So Breslow published the comparison of the hexatriene with respect to benzene in 1962, if I remember well, okay? And so, of course, opening the ring of PAC gives a trianac. So now the trianac can be saturated at one double bond to give the dianac. And finally, it can be saturated again to give the enac. And the enac, if it is aromatized by considering the triple bond as a 2-aniline, it gives back to the back. So it is kind of cyclic device, okay, for the optimization of the pharmacophoric unit. As you can see, of course, if the aromatization of the dianac could give a cyclobutadiene derivative, which would be highly unstable, so it is not worthwhile to test this way. So finally, as you can see here, so actually the idea was to target the enac by what we call the small symptom strategy. Actually, when you consider the DAC derivatives, okay, and the PAC derivatives, you can separate the structure into a unique chiral DAC pharmacophore on the left and a versatile achiral lipidic oxophore on the right, okay? And if it was possible to couple in a systematic manner, so the fragile resolvable C5 nucleophile with a standard CN electrophile with N greater than 10, okay, then it would give a general route to various DAC and to with a view to structure activity to optimizing the structure activity relationship. Actually, the small symptom, namely the C5 DAC, which is depicted here, can be resolved by enzymatic kinetic resolution. That was done by Dimitri Listunov in 2019, okay, it was published, and then that was optimized by the Conceição Ferreira de Oliveira, a collaborator in Brazil. And as you can see, the small DAC, okay, the C5 DAC can be obtained in optically pure form under the R configuration, being the eutomeric form. So that is a key in the design of the structure activity, of the development of structure activity studies. So, but first, okay, sorry, I will come back, because the main question is, does the small enantiopure RC5 DAC resist to reaction conditions with electrophile, both stereochemically, but already chemically? So first, we envisage to make the substrate react with various electrophile as a nucleophile to various electrophile. So it is very formal. First of all, of course, trying sonogashira coupling with iodobenzene, okay, gives a corresponding pack, okay, it is a model reaction, showing that actually the yield is not so good, it is only 45%, but the DAC is compatible with sonogashira coupling, giving the non-lipidic back, so in 45%, as I mentioned. So with respect to carbaldehyde, to keto electrophile or carbonyl electrophile, like the dodecanel, which is shown here, actually the DAC diol was obtained with a moderate yield, a quite low yield. Actually, it is namely 22%. So finally, the small symptom actually can react as a nucleophile to a electrophile. Also, the reaction condition must be optimized to improve the yield. Coming back now, what is that? Okay. Coming back to the NAC target, okay, it could be reacted, the substrate could be reacted with bromopolyane, alkene, with a terminal bromoalkene, okay, which was obtained by hydroillumination and oxidation with N-bromosuccinimide, okay, and after coupling, the corresponding NAC could be obtained. One silicon group has disappeared at the central structure here. The TIPS molecule, the TIPS fragment has disappeared from on the left here. But finally, it could be deprotected, and the corresponding NACs with two different chain lengths, okay, were obtained. And as you can see, the cytotoxicity against HCT116 cancer cell was fantastic, okay? 42 nanomolar in the racemic series, okay? And by comparison to the DACs, to the corresponding DACs, okay, the unsaturation, okay, adding an unsaturation in the DAC, again, improved the activity by a factor of 5. So that is the fifth improvement by a factor of 5 by structural optimization. Of course, now the NACs are very promising, but are not obtained yet in the optically pure form. Namely, I will go to the conclusion, okay, and the prospect about that, about the lax anti-tumor activity. Regarding chemistry, of course, it can be continued. And after the natural AAC, then artificial DAC, then the fragile BACs and the stable BACs, and now we have been led to the NAC. of course, in the racemic form, but what about the optically active, optically pure SNAC target? And of course, the next step is an urgent step, namely the DIANAC target, which could be less active at the level below 1 nanomolar. Okay. Of course, in biology, so this is for chemistry, so in vivo studies must be pursued, especially regarding pharmacokinetics and ADMET studies, okay, and possibly preclinical tests on xenograft murine models. So, but first of all, we need to select a lead. So, of course, the outline of my next talk at the Kharkiv seminar, I don't know, it can be in a few years, will concern the two other parts, namely the mycobacterial activity of the LAGs and the anti-cancer dacamines. So now I would like to come to the acknowledgement section. And I would like, so all the people whose name are in boldface are provided the main results which have been presented today. Other people have contributed, but I would like to emphasize those people. And I would like to emphasize especially Viviane Perrou, Dunia El Arfaoui, Dimitri Listounov, Margot Bossua, François-Xavier Toublet, and Sébastien Briton, and other people, okay? However, I would like to highlight the role of Dimitri Listounov, okay? And Dimitri Listounov, who was a doctorand and then a PhD student and then a postdoctoral fellow in my group, and did open all the prospects that of the chemistry we have developed here. So his name is very important. I would like to thank especially Etienne Joly, who was very enthusiastic with this project from the very beginning after the first development. And also, of course, Zoya Voitenko from Taras Shevchenko University in Kiev for co-supervising Dimitri Listounov during his PhD in Toulouse. I would like, of course, to thank Julian Volovenko, her colleague, and the CNRS French-Ukrainian GDRI and the French Embassy in Kiev. I would like now to acknowledge the Ukrainian Science Prize, because this is my diploma of Dr. Honoris Causa, and here is the medal, the copy of the medal for the centennial of the Academy of Science of Ukraine that I received. I was awarded in 2019 as Valentin reminded, okay? And okay. Now I would like to advertise for Chinese Chemical Letters, of which I am an editor, okay, European editor, and finish with the French-Ukrainian Journal of Chemistry, with special thanks to Igor Levkov. I haven't had many news from him for now a few months. However, he's very brave, continuing to edit the journal in the condition today. Then I cannot stop my talk without evoking, of course, in Kharkiv, because I am in Kharkiv, the name of Oleg Shishkin, who was my first contact in ESC, okay, of course, and of course I was introduced to Valentin later, okay, very after. Of course, it was Svitlana Shishkina, and also, because we are in Kharkiv, Sasha Rochal from the Karazin University. And I would like to, I cannot finish, of course, I cannot mention all the people in Ukraine, of which I think every day, but I would like to have a special thought of Dmitro Surov, okay, who is currently a PhD student with me in Toulouse. And of course, he is very brave, and now he is in Kiev in difficult condition, and I hope he can come back someday. So I thank you for your attention. Sorry for having been 5 minutes longer. Okay, sorry for that. And I am ready to answer any question. And of course, it was very, very, very dense. Okay, so we can send you the presentation and the detail. Thank you very much.
Thank you very much for your nice lecture. By the way, Svetlana Shishkina and Alexander Rashal is here with us on the seminar.
Fantastic.
Yes. So, dear colleagues, do you have questions for our lecture concerning this nice chemistry and biological activity of these compounds?
Yeah, of course. I'm a chemist, but I'm not very, of course, I am very, very excited by the biology because, of course, my colleague Etienne Joly and then Sébastien Riton did a lot of work on that. So, but I can try to answer any question, whatever the, the topics.
But at the moment, I don't see any hands raised.
No, I know it is, it was complicated for me because the historical part is important. I cannot go directly to the latest result. For instance, the ENAC story I finished with, okay, is the end of a story. It's not the end of a story, but it is a very latest development, but it came naturally from the previous story. So that's why it is complicated. It is quite busy, a busy presentation, I know it was.
Thierry, do you want to ask?
Yes, I had a question regarding the asymmetric addition on the acetylene on the aldehyde. Did you sometimes try to optimize the structure of the binyl? Because you're using Carrera's method, and sometimes you'll find in the literature the 3,3' dibromo binyl instead of binyl. Is it something you tried?
No. Actually, we have tried to optimize the binyl by another method. It was not tribromo. Actually, it was, if I remember, it was Afida Gaspar who devised the fluorinated binyl system. Okay, no, sulfonated binyl system. And actually, we could not optimize the enantiomeric excess by this way, but from the Spus method. Okay, but of course, it has been envisaged. Yes, we did that.
Okay, thank you.
But if you want actually. It was envisaged and it was even published, I think. I can send you the paper, but there is no improvement marked, big improvement. And actually, at the end, N-methylephedrine is cheaper than binyl, okay? And finally, we focused more on for the dialkynyl carbinol, okay, with 2 triple bonds, on the modified Carrera's method. And of course, the Pous method could be developed, okay, for also other AAC, just for comparison, for the sake of comparison, not for the sake of absolute activity, improving the absolute activity, but just for that, it was very important.
Okay, good. Thank you very much. Thank you.
Thank you.
Somebody else?
Of course, I'm ready to answer any question by email. Okay, if you have any question, you have my email at the very beginning of the slide, and there is no problem for that.
Yes. Concerning your further plans and this dearomatization process and improving bioactivity, do you have, it's only idea, or you have a molecular mode docking for this theory?
What do you mean, molecular?
Dearomatization of your pack, and you are going to improve your biological activity. And
yes,
it's only theory, or you have a molecular docking for this?
No, it's just theory, just pure theory. No, it was actually, of course, the dearomatization cannot be performed chemically. Of course, it is very conceptual, okay? But also, there is no, what I would like to illustrate is that that was the basic idea behind the proposition. if you have several domains, so in the group, we had several domains of application of our molecules, organic molecules. And finally, for medicinal chemistry, perhaps, you know, the theory of aromaticity on which we work, especially with Igor Komarov, okay, because of because of the I would say graph theoretical appraisal of the activity of the structure of aromatic system and conjugated system, actually, which can be refined by DFT calculation, but even at the level of basic topology, Euclid theory level, okay, we can have a lot of information. Of course, it's not sufficient. I know, I know. No, we don't have any calculation, but you're right. You're right. We should try. We should try to put. We should try to put the ENAC and the DIENAC, and why not the TriENAC in the receptor pocket for the HSD 17B11 receptor. Yes, of course. Protein. Yes, you're right. We should do that. Of course. We were focused on very abstract theory, and then experimental science, mixing, you know, nucleophile and electrophile. But you're right, docking with, yeah, I think, yeah, that would be very good. Yeah, I hope some of my collaborators can listen to this presentation and will benefit from your suggestion to perform the calculation. Of course.
Yeah, okay, we will share.
Frédéric Rodriguez could. Yeah.
We will share this video in our YouTube channel, and maybe some additional scientists from Europe or from China or other countries will contact you about the continuation of this topic as well.
Oh, thank you very much. Thank you very much. Of course, it's not complete, but I'm completely ready to answer and to discuss, and of course, by email. No problem for that. Thank you very much for providing the lecture on YouTube. Okay, that will be great.
Okay.
Even if it was not perfect, because I had this, but anyway, you know, I'm like an actor. We are all, you know, when you give a talk, you're like a class for the student. You are an actor, but you don't have the right to do it several times and to say the only one. Okay, but it's great. It's spontaneous. Thank you very much, Valentin. It's a fantastic experience today for me. And I would like to, I don't see all the audience, all the people. I would like to say hello to everybody. I know I don't want to mention names because I may forget some, but hello to everybody.
Okay.
I know. Yeah,
so,
I know.
Dear Remi, thank you very much for a very interesting lecture. I hope you will give another lecture, maybe, yeah, you said about this. In the future on the Kharkiv Chemical Seminar, and I hope this seminar will be exactly in Kharkiv, not online in Zoom. And
I hope so.
For us.
Of course. I support all of you in those conditions. I will not cry, but because I know you're brave and you wouldn't like that. But however, I support all of you, and with my heart. Okay, this is very important.
Thank you.
Thank you to you and to you all.